Chapter 4, Infection, Infectious Diseases, and Epidemiology. The overview for this chapter is we're going to be covering symbiotic relationships between microbes and their hosts, reservoirs of infectious disease of humans, the movement of microbes into hosts, infection, the nature of infectious disease, movement of microbes out of hosts, portals of exit, modes of infectious disease transmission, classification of infectious diseases, and epidemiology of infectious diseases. So let's launch right into the first topic, symbiotic relationships between microbes and their hosts, Chapter 14. The learning objectives for this topic are distinguish among the types of symbiosis, listing them in order from most beneficial to harmful to the host. Describe the relationships among the terms parasite, host, and pathogen. Describe normal microbiota, including resident and transient members. Describe 3 conditions that create opportunities for normal microbiota to cause disease. An overview of this topic would be, we're going to cover symbiosis in general. We're going to talk about the types of symbiosis, normal microbiota and hosts, and opportunistic pathogens. First off, there are microbes all over the place. They're in the soil, they're in the air, they're in the water, they're on you, they're in you, and very few either harm or benefit us. Other types of microbes live in close association with us, and that's what we're going to be talking about in this topic. Symbiosis is the term that we use to describe any situation in which microbes are living in close association with either other microbes or with multicellular organisms, and it is used to describe everything from beneficial to harmful interactions. So let's go over the types of symbiosis, and let's start out with the most beneficial to both organisms. Mutualism is where a microbe benefits the host and the host benefits the microbe. Termites are an example of this. Now remember back to Chapter 12 when I told you about Trichonympha? Trichonympha lives in a mutualistic symbiosis with termites. They enable the termites to digest the cellulose that they're eating. The termites keep the Trichonympha nice and warm and moist and provide them with regular food. Both parties benefit. The next type of symbiosis is commensalism. That's where the microbe benefits, but the host is neither benefited nor harmed. Now, the book uses the example of Staphylococcus epidermidis, our most commonly isolated skin bacteria from humans. They benefit from being on us. We provide them with food, and they don't harm us. Now, on to the harmful relationships. Parasitism, where the microbe benefits, but the host does not. All diseases are an example of parasitism. Some parts of humans are enzonic. In other words, the only cells in your blood had better be just your cells. Same with your brain, anything that's on the inside of you. But anything that's technically on the outside view, in contact with the environment, is home to all sorts of commensal and mutualistic organisms. So, for example, the skin has approximately 3 million bacteria per square centimeter. That may seem like a lot, but when you go to the large intestines, where you have 400 to 1,000 kinds of bacteria, and they're outnumbering your human intestinal cells, the skin is relatively free of bacteria. The difference being, the skin is dry, your intestines are nice and moist and warm. Now, there's 2 types of normal microbiota. There are the resident microbiota. In other words, if I were to sample your skin or your intestines, then I would find the same sorts of organisms all of the time. They are resident. Technically, if they are found to be present for longer than 6 months, they're considered to be resident. Organisms that are just passing through are called transient, if they're there for less than 6 months. Usually less than that. So, for example, if I were to swab your hands, I would consider the staph epi that I isolated to be resident normal microbiota of your skin, but if I found any E. coli, I would know that they were transient microbiota of your skin. Now the reverse would be true if I were taking a fecal sample and culturing that. You begin to acquire your normal microbiota as soon as you're born. You pass through the birth canal, you're handled by your parents, you pick up their skin bacteria. As you're breastfed or fed by a bottle, you start picking up their intestinal bacteria through transient microbiota on their hands and such. Normal microbiota do not cause disease under normal circumstances. They're there to help you out, or they're there and not harming anything. But if you are immunosuppressed, patients who have AIDS, patients who are undergoing cancer treatment, patients who have had an organ transplant are immunosuppressed, and that allows normal microbiota to expand beyond what is healthy. Also changes in normal microbiota. Infection with Clostridium difficile is an example of this. If taking antibiotics lowers the numbers of normal microbiota that are either mutualistic or commensal, then it allows some opportunistic pathogens to expand beyond what is healthy. Also, if you introduce normal microbiota into an unusual site in the body. So in other words, if I get skin bacteria into my blood system, that can cause a disease. Your immune system usually takes care of it. Yeah, but I've used the example of getting skin bacteria on implants, hip implants, knee implants, and that causes disease. That's it for this topic. Here are reminders of what the learning objectives for this topic are.